schon drin.
Ja.
Mom and Dad taught us being Murphy makes us special, but that's a little harder to believe now that I'm in high school.
Hi, Boze.
Good evening. How are we?
Fine. How are you? Welcome.
Very good. Thank you.
Boze, this is Cody Carroll, who's one of our interns. Cody Carroll.
Hi.
Hi,
Nice to-
Cody. And a few folk gathering.
Where are you joining us from, Boze?
I'm in Rhode Island.
Nice.
Yeah, it's not too bad. Jamestown Island in Narragansett Bay. So
That's a
very-
nice area.
pretty
I used
spot.
I used to sail intercollegiately, and we sailed a lot around there.
Right. Yeah. Between Newport and Jamestown, no doubt.
Yeah, exactly. Good evening, everyone. This is Boze Hancock. There's William. William is a diver with, he's done a lot of diving with Mike Carew, who you've met, Boze. He's he runs a dive shop on City Island. And William is one of his students, and Ellen
Right.
Grace.
Hi,
They do
Ellen.
a lot of monitoring of
How are you?
oysters. And they say Eddie Spallina, who's the Commodore of one of our local yacht clubs.
Hi. Excellent.
Hi, Cody.
Oops. Sorry, that's
Hi, how are you?
Good. That was a great presentation, Cody.
Oh, thank you.
Really excellent.
Boze, Cody did a presentation on we have ORS cages from the Billion Oyster Project, and she and others have been monitoring their growth, and she did a presentation to the volunteers and the board, and it was excellent.
right.
Ellen Grace
Yeah.
is one of our superstar, amazing volunteers. Yes, you are.
True.
Don't be modest. You are.
It is absolutely true.
Yeah.
It's such a great, great, great grassroots, wonderful organization. I love it. Thank you.
Yeah, I saw you guys' Facebook page. You guys do excellent work out there. I see your pictures. You guys are the best out there. I can see it. But sadly, I couldn't come to any of your events because I was fortunately busy with my college semester at Westchester, finishing off my associates, and then I was also working at an animal rescue facility in Walden, New York, Orange County last fall. That's why I couldn't come, but I hope to join you guys, whatever you got this year.
Definitely. We have a lot coming up, William. We're going to get you involved. The shell collection is going to start up again. You might be interested in that.
Yeah,
Fine.
definitely need to get out again because last year, crazy year. Because I locked in again for almost a month. Almost a month in my house. So
Yeah, it's been tough being all locked inside. Here's Katie Mosher from BOP. We have people from all over.
Katie, it's been quite a while.
Hello. Nice to see you, Boze.
Yes, and yourself. Fantastic.
Katie, thanks again for meeting with us the other day. It was so helpful.
Oh, my pleasure. Anytime.
Really appreciate it. Cody did an excellent job making her presentation on the data.
Yeah.
It's hard to pull all that information together for people who are not scientists and to make it an understandable narrative. And she did that. It was really great.
Yeah, it was really well done, Cody.
Yeah.
Thank you so much. I had a lot of help.
yeah, it's also crazy is this semester in my college classes chemistry was always the hardest for me. I did not like that class at Westchester because I honestly failed it.
You have to take it again then.
Yeah, I'm gonna do that. Do that in the fall.
Good. Maybe
Fall,
there's someone who can help you out, someone, like, a friend or someone in the
yeah,
family
yeah,
can help you out.
because a lot of math, which is obviously crazy. Other class, yeah, chemistry. Everyone calls it the hardest course I ever taken in college. So I'm not the only one who failed. The others.
I have faith in you, William. We just, Rob Kraft, Boze, I don't know if you know Rob Kraft, he's from SUNY Maritime, and he's very involved in oyster projects in his hometown and also through SUNY Maritime.
Yes, we've communicated.
You're getting a great turnout. We're at 28 people already. Fantastic.
Hey, you.
Eric Sanderson, he's one of our advisors. He's just getting on too. Hi, Eric.
Hey, Sally, how you doing?
Fine, thanks. How are you? Thanks
Good.
for joining us.
I'm looking forward to it. Thanks for the invitation.
You're very welcome. We'll get going in just a little bit. We still have people coming on. We have a lot of questions, Boze, about your project SOAR.
Right.
Once we
Yes.
get to the question and answer, it's fascinating what you've done with not wasting the oysters and using them.
Yes, it's been a fun project, that one. Actually, it hasn't had a downside so far, which is amazing. Been a lot of activity and everybody's happy. So, yeah, it's been a fun project to be a part of.
Fantastic. Well, we're getting people. I think we're pretty settled in. We have 33 people on board right now. So why don't we get started? We are thrilled to have Dr. Boze Hancock present to us tonight, a webinar on shellfish restoration one-on-one. It's really my honor to introduce him. Dr. Hancock is a senior marine habitat restoration scientist at the Nature Conservancy and is a member of their global marine team. He got into the field of bivalve restoration in response to a massive oil spill off the coast of Rhode Island many years ago. In 2004, he ran one of our nation's biggest shellfish restoration projects for NOAA and for the Nature Conservancy. Since then, Dr. Hancock has been active all over the world promoting and developing oyster shellfish restoration far and wide and locally. He's been in the Asian Pacific region in Europe, United Kingdom, Australia, here in Chesapeake Bay, Puget Sound in Washington, in the Gulf of Mexico, in the Great Bay in New Hampshire, also as far and wide as Hong Kong and New Zealand. During COVID, he's been instrumental in creating a project called SOAR with the Nature Conservancy. And basically, due to COVID, as you know, many restaurants had to close and weren't selling the shellfish they typically do. And so he and others purchased over 5 million live oysters and put them basically back into the ocean and the sound, and have into restoration, oyster restoration reefs throughout the country. When I reached out to Dr. Hancock, I really thought it was a shot in the dark that we would get him, because he is a world-renowned figure, a world-renowned scientist. But instead, he listened very closely to the work of the City Island Oyster Reef and what we've been doing for the past year. And he was tremendously supportive and really thrilled to be part of this community-based partnership working for a cleaner marine environment. Dr. Hancock also has an extensive resume of research and writing, and has developed the concept of an oyster calculator, which determines how much oyster restoration is needed in a particular area. After tonight's webinar, I'll send to everyone on the call recommended publications and scientific articles that Dr. Hancock had recommended. After his presentation, which will be about 40 minutes, he'll take questions, and please mute yourself during the presentation unless you're asking a question or making a comment at the end. And now I will turn it over to you, Dr. Hancock. Thank you so much, and CIR welcomes you warmly.
Wow. Whoa, Sally, thank you very much. That's pretty amazing, actually. That's a lot more information than I gave you. So you have done quite some background research. Thank you. And yeah, happy to talk to this group. This takes me back to my roots. Working with community groups to get things to happen is just great fun and hugely productive. So I will share my screen and see if I can get a presentation going. Now, there we go. That is hopefully looking right. And if I have a fault in presentations, and I should be reasonably practiced at these by now, but there's just so much to talk about. So I will try and whip through in 40 minutes. If I jump a slide or two, it's because I'm running behind time. But yeah, this was just an opportunity to really dive into the logic of shellfish restoration and try and walk through you know, from beginning to end, what the logic is and how it's done. So I hope I'm pitching it at about the right place. In overview, I've broken this kind of into 4 different topics. You know, why invest, what it looks like when we're implementing, translating some of the science into usable information, and opening up some of those enabling conditions, decision support, and the oyster calculator, which Sally mentioned. So very briefly to start with, you know, why habitat. I'll just go quickly through some of the loss, the history of restoration itself, and ecosystem services. So this map, I imagine many people are familiar with, actually. This is one that we produced in 2011 from a report that was actually produced earlier. So it's over a decade old. But what this was an attempt to have a look at the condition of shellfish habitats. So the shellfish reef as habitat globally. All of those little round dots are bays where we could compare the current amount of oyster habitat to a historic amount. And that historic amount was generally between mid 1800s and early 1900s. So about 100 to 150 years. And the take-home message from this is that those bays and estuaries were averaged up to ecoregions, and the dark red represents ecoregions where the average condition contained less than 1% of the historic amount of oyster reef. The lighter red is less than 10%. So kind of in summary, globally, we had less than 15% of oyster reef left, which is by far the lowest of any of the marine habitats, the critical coastal ecosystems anyway, that have been measured. And that's a very conservative estimate. To get this information published, we had to be conservative. Most places, it's less again. So this is a publication looking at the US in a kind of finer view.
Sound you made me play.
Oh, is that Carl? Carl, you might mute your microphone there. We're hearing everything in the background.
Not right now.
Alina and Carl. So, I mean, this study actually had managed to have a look at biomass as well as area, and we're down to 12% remaining in terms of biomass. The UK, if we open that up, we've got less than 1% there. South Australia had an excellent study done, also less than 1% remaining. Regionally across Southern Australia, we're down below 1%. On the East Coast with the different species, up around 8%. And we are currently undertaking that kind of historical ecology view of where we are in Hong Kong and China. So really there's not a lot of this habitat left, is the bottom line from that assessment. And one of the things that I like to point out is that when we're thinking about shellfish, I find an easy way of describing why shellfish are important is to point out that they are ecologically the equivalent of a coral reef in the tropical systems. And the corals are the reef builders in clean tropical water. As you move towards the poles and on either side of the equator, it's the shellfish that take over that ecological function, or should do if they were left, if there are any still there to do it. So they are exactly the ecological equivalents. And a couple of extra points. One is it's not just oysters. Mussels and oysters are by far the most important habitat formers, but there are some others as well. At top left there is a pinna shell in South Australia, and on the right here is a pearl oyster, a small pearl oyster species, having a similar function in the habitat. And the other point there is that it's shellfish should actually be one habitat within our coastal systems, but we don't have time to open up the other structuring habitats. But we have had some practice at putting oysters particularly back again. I'm involved in mussel restoration, particularly in Australia and New Zealand, but we've had most practice with oysters. This map is a representation of just the work that TNC and NOAA have done. That's over 300 projects between us, and not only is that information a little out of date, but that's not all. There are certainly more projects than that. So in America, this work is well progressed, and we've got a really good idea of how to go about it. And I've been lucky enough in the last 6 years or so to be working deliberately to take some of that learning that's been done in the US and take that out of the US and start oyster restoration programs in China, Hong Kong, Australia, New Zealand, and the work in Europe is really gaining momentum at the moment. So there is a growing understanding of the importance of oyster reefs, which leads us to the question of, why spend money putting this stuff back? It's gone, so what? And the obvious answer to that is because of the ecosystem services that habitat provides. It's because of what it gives back, what we gain. And these categories come from the Millennium Assessment. I won't talk that much about the cultural value that we get. The aquaculture and fisheries product is something that we're all familiar with. I tend to talk more about the coastal protection and the water quality, the denitrification, filtration, and also fish production. And of course, we have learnt repeatedly, actually, over the last 10 years, that if you want to create jobs, this marine habitat restoration is actually one of the best ways of going about doing that. So I'll just touch on quickly some of those main ecosystem services. And the first is coastal defence. We think about that in two different ways. The first is as a submerged breakwater, and you can design a reef that can be designed to have the oysters as the biological covering of a reef. and the design can be such that it causes waves to crest and break and spill their energy. Pretty obvious that's how breakwaters work. What is less obvious but really important is the friction that these structuring habitats, not just oysters, but all of our structuring habitats create. Waves touch the bottom at 7 times the wave length, if you remember back to your physics classes. And as soon as they start to touch the bottom, the friction coefficient of the surface that they're rolling over has a big impact on how much energy is dragged out of that wave from underneath. So maintaining oyster reef, tide marsh, seagrass, some of these structuring habitats really has an impact on that friction coefficient. And here's a couple of photos. This is one of the bigger shoreline protection projects. This is from Mobile Bay, Alabama. On the left there, you can see what turns out to be 3.5 miles, I think, from memory, of protecting reef. And on the right there, a close-up where you can see that reef designed as a breakwater is slowing water, it's trapping sediment behind, it's causing deltas of sediment to build up, and that salt marsh is actually growing back out again from what was an eroding reef to come out and meet the barrier. If we look at water quality, that can easily break down into the filtration, direct filtration effects and denitrification. And if we're talking about direct filtration, this graph is meant to represent on the y-axis over here, the vertical axis, how many days it would take for all of the water in a bay or estuary to be filtered through an oyster. And we can compare that here on the x-axis to the amount of time, also in days, that water on average would stay in a bay, the residence time of the water. And an example here I've got is Chesapeake Bay, where the residence time's about 30 days. And the volume of Chesapeake Bay is enormous. But the calculations that were done quite early on, actually, back in about 04, I think, were that historically, the oysters in Chesapeake Bay would have filtered the entire volume of that bay in between 3 and 4 days. And with what we've done to the abundance of oysters, that's now up at about 400 days, so it's over a year. And if the water stays in that system for 30 days, then this diagonal line represents where the oysters are having the dominant impact on water quality. It's not a matter of flushing dirty water out to the sea and replacing it with other cleaner, hopefully, water. But the work is being done internally within the residence time of that water in the bay. So that's one measure. And what they're actually doing when they're filtering is taking all of the particulates out. You know, oysters have this neat habit of taking out pretty much all of the particles from the water column. What they want to eat goes in, that gets ingested. What they're not interested in eating gets attached to a mucus thread and spat out. It's heavy, it sinks down into the reef. And you can measure that filtration impact. This photo has fluorometers. To these gadgets in the water are fluorometers. They measure particle concentrations in the water. In between those two gadgets is an oyster reef that we know the density, the biomass of oysters, and you can plot out the concentration of particles upstream and downstream. So that's the reduction in the stuff, the muck in the water from water flowing across that reef just one time. So you can really measure in the field the physical impact of that filtration. The other really big impact on water quality is denitrification. So organically available nitrogen is the nutrient that's responsible for the phytoplankton blooms. That's the one that really ramps up the phytoplankton. That's what blooms, dies, sinks to the bottom. The degradation of that, the bacteria breaking it down, sucks all the oxygen out of the water, and you wind up with anoxic zones, fish kills, and the problems that we've seen and you're all probably familiar with at your end of Long Island Sound. And it turns out that oysters or oyster reefs don't do much of the denitrification themselves, but they facilitate the highest rates of denitrification that we've been able to measure. And what that is done by the bacteria. The bacteria that are responsible live on the boundary between oxygenated and anoxic layers. And they occur in the shells of oysters, they occur in the gut of oysters, but most of them are in the sediment. And instead of making their living by metabolizing oxygen to produce energy, they actually break down ammonia, nitrate, nitrite, those nutrients that cause the phytoplankton blooms, and break them down into dinitrogen gas. 78% of what we're breathing now is dinitrogen gas. It's inert, it's out of the system and gone for good. And it's one of the only ways we've got of actually permanently removing that nitrogen from the system. And when we have a look at a few results, this top graph, the red bars are from an oyster reef, the amount of nitrogen removed from an oyster reef. The blue is the amount of nitrogen removed from a control, so the sediment that was there before a reef was restored. And you can clearly see the magnitude of that result. The bottom study is one that was done in Rhode Island, just comparing oyster reef with some of the rack and bag aquaculture. Aquaculture has a very different denitrification signal depending on the kind of aquaculture. But in this case, with rack and bag quite close to the sediment itself, the signal was pretty high and much bigger than just shell by itself or control. The other thing that really drives a lot of the benefit, a lot of these ecosystem services from oyster reef is the biodiversity and the biomass that they produce. So on the left here of this table, we've got just groups of animals, the amphipods, anemones, barnacles, clams, you know, the different groups that you can measure, with unrestored in the left two columns and restored in the right. And whether you're looking at the number of animals, abundance, or the weight of those animals, the biomass, you just run your eye across those columns and you've got a 1 to 3 orders of magnitude increase in both biomass and biodiversity from the oyster reef compared to what is basically a mud or a sand bottom as a control. And that's really important in fish production. You know, the way I describe an oyster reef from the perspective of a juvenile fish, a baby fish, is a nice secure house with a well-stocked fridge. You know, all of that biodiversity is essentially fish food. These are the really small crustaceans, the polychaete worms, the things that small fish love to eat. And in that reef is a nice secure home. They're protected from a lot of predation. And lo and behold, survival goes up. And when the survival of the babies goes up, you get more big fish. And this is the result of a study to actually measure the amount of fish that are produced from an oyster reef in our kind of latitude, mid-Atlantic. On the y-axis here, this is easiest read in tons per hectare. So this is 3 tons per hectare production from the Atlantic. And on the x-axis is years. So 3 things to take away from this graph. One is 3 tons per hectare is a lot of fish. The second, if you have a look at how fast they accumulate, you actually get to full production quite fast. And there's some really nice work coming out of the coast side of Chesapeake Bay, so on the Atlantic, Virginia Coast Reserve, where 6 to 7 years seems to be the point at which a restored reef reaches its full potential in terms of ecosystem services. And the third point here is that there's some interesting species. Some of the snappers, gropers, pigfish, sheep's head get people's attention. And this comes actually from the last economic downturn, not the COVID pandemic, but the housing bubble and the ARA or Obama stimulus work that was done, where we've actually managed to measure the job creation from some of the natural infrastructure and oyster reef, marine debris removal, fish passage. These are all very high job creators. So in terms of blue economy, it's a very powerful impact. So I am behind. I'll have to get cracking here, but I did want to just show some of the types of restoration and a bit of a history, starting with some of the earlier projects. But first up, there's a decision to be made at the very beginning of considering any restoration work, which is, do you need substrate? Do you need to build your breeding population? Are there larvae in the system? Is recruitment high enough to sustain a reef, or do you need to build the breeding population? And it's much easier if there are larvae. You know, in that case, we only need to add structure. If there aren't sufficient larvae, then we need to back up to the hatcheries and produce the oysters to go out on that reef to build a population to provide those larvae. And there's a number of different substrates. I'll run through a number of different scales, but if you don't have to actually build a breeding population in order to provide those larvae, you go straight into just 2 to 3 years time measuring you know, quite a mature collection of oysters that are by 3 years breeding well. And if you are not so lucky, then it's back to the hatchery, producing larvae, setting those larvae onto something. They tend to get set onto shell, and we talk about spat on shell. You know, this scales up so that the spat on shell can be bags in a tank, such as that photo, or the kind of scale they're using in Chesapeake Bay, where there are huge baskets that go into these setting tanks on a jetty. You know, bags can get grown out in aquaculture facilities and spread by hand. Those large setting cages get emptied onto boats and hosed off in large volumes. So there's a number of different scales that we need to think about. But underlying all of that is your topography. Height matters. Every time this has been looked at, we've come to the same conclusion. You can't just put oysters straight on the bottom. They don't do very well. You actually do need to recreate that reef structure that's been destroyed before either allowing recruitment onto that structure to build your biology, or seeding oysters to build your biology. And there's been some great work done also in the Chesapeake, looking at about 12 inches, actually, about 30 centimeters, as being a tipping point at that particular latitude of where a reef grows and thrives, or slowly degrades and disappears. So what do some of these projects look like? Here's one from Rhode Island, very community-driven projects, the kind of things you might be thinking about. You know, weathering shell, bagging shell, getting that out into a reef format. There's a growing number of examples of pretty large scale work where we've got, you know, 200 to 500 ton barges deploying material. Some of these are older projects going out. The South Carolina projects tended to be about a decade plus old now, and this is just building up enough of that substrate in the intertidal, where you can change the objective of what you're doing and have different objectives. This is shoreline protection work, also done on a fairly small scale, building up that reef in front of eroding salt marsh to protect the toe of the salt marsh, so that both habitats are then working for us. And a number of different substrates have been tested and used with good results. It's not a one size fits all. It really is a matter of thinking through what you want to do, what the oyster reef is intended to provide, and choosing substrates accordingly. This photo is from Tampa Bay, where this is a part of a shoreline protection project. There were a number of different configurations of some of these cement structures tested, and you can actually see here the delta of sand behind that barrier that's building up. This was a heavily eroding beach area, and you can see that after a few years, the sediment has filled in this gap between your barrier and the beach, and you've got salt marsh regenerating. So these can be very successful. These are shots from actually the area of Virginia Coast Reserve, the Atlantic side of Chesapeake, where they're using some of these oyster castles, overgrown Lego blocks. Actually, you can lock them together and build structures of different shapes. And in that situation, they've got fantastic recruitment, a lot of larvae in the system. And after 3 years, they have basically struggled to find any of the concrete that's underneath that creates the substrate for that reef. It's enough to make most people very jealous. So these are built for very different purposes. This is a shot of a couple of different reefs actually that are being put into Galveston Bay in Texas. Texas does not yet have legislation or regulations that control their fishes. They can't close areas to fishing. So in Texas, if you're spending public or private money actually restoring a reef for ecosystem services, you can't exclude the fishes that will come and take the oysters and reverse all of the good work you've done. So what they do there is build their sanctuary reefs, as they call them, out of rock that is too big to dredge over. You can't run a dredge over the rock on the left there. They are also building reefs for harvest and using much smaller rocks. So that's a nice example of just thinking laterally. In the sanctuary rock, if you look at a side scan sonar and build up an image of what's gone into the water, the sanctuary reefs look very different, and they're not the kind of thing a skipper will tow a dredge across. And of course, different scales. We've got a number of different scales to work with at the moment. The left is Indian River Lagoon in Florida, where boat wakes have been causing oysters to dislodge and build up above the waterline. And this is a group that are restoring the reef below the waterline again, compared to a project in South Australia where this is, I think it was 2,000 tons of substrate going in to build reefs. On the side of that barge is what they've called their cake tin. And the closest loader there is dropping rock into that, essentially a mold that goes down 6 meters into the water. It's only about 4 meters off the bottom of the ocean at that point. And by dumping your rock into that, you can quite precisely build reefs where you want them, in amongst other habitats on the bottom. So there's a quick overview of implementation, hopefully from a number of different places, give you some ideas of what's going on around the world. We've also put quite a lot of work into not just developing the science behind these quantifications, the fish production, the denitrification, but trying to translate that science into usable documentation for project managers, regulators, the government departments, those kinds of things. And this spans quite a time period at this point. Best management practices started in 2006. This one's from 2011. It's actually an interstate shellfish sanitation conference best management practice, which includes restoration, which is interesting. This is the international version that came out last year, published by a global group really just walking through some of this, how to start thinking about shellfish restoration. That's actually been translated into Mandarin and Cantonese, and we're looking to translate into Spanish. There's a monitoring handbook, which is going through a few details that I'll open up in a second, actually, and a manager's guide, which walks through some of these ecosystem services and the online oyster calculator that Sally mentioned at the beginning. So monitoring, just briefly, I'm gathering that you guys are quite expert at this monitoring. So very briefly running through some of the how-to that's encapsulated in that monitoring handbook. And really the idea there was to come up with a basic level of monitoring that would allow us to compare results between projects. It was getting very frustrating up to about 2014 when this was published, that each project was so independent we couldn't actually compare results. And just by getting 4 basic metrics, an aerial extent, a reef height, and then a look at what the oysters are doing, essentially in size and number, density or size frequency. Just those 4 elements allow us to give a fairly deep, actually a surprisingly deep comparison of results between projects. And if we pair that with some environmental variables that should be available to any project, just basic temperature, salinity, and dissolved oxygen, those universal metrics, as we've called them, are the ones that we are trying to promote all projects to get. And if we can get that basic level of information, then we have a fairly powerful comparison. And then there are all sorts of different levels that different projects can go to, depending on the objective of the project, what it is you want to achieve from it. And these tend to go quite quickly into measuring some of the ecosystem services. One that I really like to add into any talk and stress to a certain extent is site suitability. All of our critical coastal ecosystems, you know, the reef systems, the oysters and the corals, and the other 3 that I tend to talk about, which is the seagrass, the salt marsh, and the mangroves, do an enormous amount for us, but they can't survive just anywhere. You need to be reasonably careful about where you're putting them and making sure that the environment is suitable. And it's not difficult. It really is just a matter of having a look at some basic parameters. Salinity, dissolved oxygen, very important, particularly the City Island. I know you've been looking at that. What's on the bottom and how that relates to what you need to build, and recruitment. Are there larvae in the system or not? I'm always dismayed when projects start that haven't done the homework, but is fairly simple. And there are a lot of other considerations as well. There's all sorts of competing uses, cultural uses, permit requirements that restrict down the amount of area available for restoration. And it's important and informative to map them out to start with. So getting on to how we try and package some of this in the decision support tools and enabling conditions. And this is where I'll just briefly open up the oyster calculator that Sally mentioned. This is an attempt to have a look at filtration and fish production in a way that we can try and picture. So this is an online tool, and it is pre-populated for all of the bays and estuaries on the East Coast and the Gulf Coast where we have information. And it comes in different sections. I'll explain very briefly. I've actually just grabbed from the website Pensacola Bay in Florida. You know, you can have a look at different bays around the coast. If we have information, it is pre-populated with the bay properties. So the bay volume, the residence time, temperature, and if we know it, and if there are oysters in the system, what kind of area of reef exists. If there is reef and there have been surveys done, we can also add in what the oysters look like on those reefs. So a density and a size distribution. We've broken that out by the sublegal and the market size, because they tend to be quite different. And what we can then do is have a look at what might happen from restoration. All of these are interactive, so this is a little sliding bar here. You can move your target. At the moment, I've got it up at what would be needed to get 100% filtration. So to meet that horizontal, sorry, the diagonal line on the graph of filtration that we saw earlier on, where is the point where oysters are filtering water? They're filtering the entire volume of the bay at the same rate that tidal flushing is exchanging that water with the ocean. And we can add in what we would be expecting from oyster restoration. And by doing that, we automatically calculate on this site what's required here. So to meet 100% filtration with this number of oysters in our restored site, that's a fairly small population, a fairly young population, we'd need a pretty big area. It's about 600 hectares, nearly 1,500 acres. And, you know, we can change all of these parameters to see how much we need to put back in order to get to that filtration level. And we also have the fish production work to this point. So we can go ahead and just tab across and automatically calculate it here for that target that we've set for 100% filtration, so the 600 hectares of oyster reef. What are we getting back in terms of fish? And this is broken down by species that were highlighted in that study. And so for Pensacola Bay, you know, you'd be looking at 860 metric tons of blue crabs produced each year from that additional oyster reef. So this starts to get into the information that can be used to discuss, you know, how much is enough. And finally, I'll just finish with some of the work that's becoming really powerful, which is getting into the cost benefit. So if we know that oyster reefs provide these benefits for us, we have an expensive restoration. What are these benefits? What's the value of those benefits? And the best of the studies, there are a number from Australia and New Zealand, but the best of them actually comes again from Chesapeake. Harris Creek is the first of the big restoration projects in Chesapeake Bay to be finished. It was started in about 2015, finished in 2018, and they're monitoring every 3 years. Sorry, it was finished in 2015 and monitoring every 3 years. So the monitoring was done in 2018. They're a big project. So the area that's shaded in the map on the left is the area of Chesapeake Bay where the restoration was actually put on the bottom. But that constitutes 142 hectares. So that's nearly 350 acres. Over 200,000 cubic meters of substrate, both shell and rock. 2.5 billion spat on shell produced from the hatchery that you've seen photos of, those big tanks, the setting tanks on the jetty, and the Oyster Recovery Partnership boat loaded with spat on shell to go and seed these particular reefs. And that restoration cost $53 million. It's an expensive exercise. But what becomes powerful is looking at what you get back. And not surprisingly, we get the two values that are pretty impressive. There's, with that, between $3 and $18 million a year of nitrogen in tons of nitrogen removed. That varies quite a bit depending on the value you place on the nitrogen. But the fishery production from that reef is about $23 million a year. So just on the strength of the fish production alone, $23 million a year is a pretty handsome return on a $53 million investment. So it starts to make some of this work look like a really attractive investment rather than just an expensive project. And so I'm getting really excited about some of the work that we are being able to describe in terms of return on investment. And I guess the final comment for any of this, which I'm sure this group is fully aware of, is that nobody does this by themselves. These are complex undertakings. They tend to be big jobs, and it does take a team. So, you know, the TNC projects I talk about and the other projects that aren't TNC projects, they're nearly all large collaborations. So I haven't gone too far over time there, and happy to talk through questions, happy to talk about the SOAR program, which I didn't think I had the time to squeeze into this presentation. But yeah, more than happy to see where we go from there.
I have a question about the measuring the larvae in the water. What methods have you used or do you recommend for getting an accurate picture of what's currently in the water?
If the aim is to get an assessment of the kind of recruitment you're likely to see on a reef, then I don't recommend looking at the larvae themselves. I recommend putting out some substrate and seeing what happens when they settle. It's a lot easier. So settlement plates are good. You know, an unglazed ceramic tile is an attractive substrate for an oyster, and that can be easily quantified in terms of area. So you've got a known area, you can get a density of recruitment. You know, if you want to replace tiles monthly, you can map out when that recruitment is happening during the season, but it requires looking at them under a microscope because they're still pretty small, you know, for that first month after settlement. If you want to make that a practical measure of what's likely to happen at a reef scale, then leave those plates there for a season, and you know, pick them up in your area in October or November, when the last recruits from September-ish are big enough to see with the naked eye, and you can then look at the size and density of the oysters that have settled over a whole season. So it depends on, you know, what your aim is again, but I would recommend going for the easy option, which is give them some substrate, let them do their thing, let them settle and grow until they're big enough to look at and see. And measure.
I have a lot of questions, but two really quick ones, and then I'll give others a chance, and then we can come back around. My first question is, Boze, are you from Australia?
Sorry, I should have said that up front. Yes, the accent is obviously not Rhode Island native. I am from Australia until 2004. 2003, I moved to the US nearly 20 years ago. Good grief! And until then, I was working in Western Australia.
I wasn't sure Australia, New Zealand. My second question is, you mentioned about height matters for the reef. City Island is a big boating, yachting community. And a lot of the negative feedback, or, you know, kind of what I hear from a lot of the boaters about our project, is that they're very concerned that we're going to grow the reef so high that it's going to affect the boats coming in and out of the marinas, and it's going to, you know, it's going to damage the boats and all of that. What can you tell us about that? Because, you know, I mean, I'm not a boater, so I personally listen to what I've heard from other people, and they said they're not going to grow that high that it's going to really affect the bottoms of the boats. But I just wanted your opinion on that.
Site suitability is where that gets kind of catalogued, and there is, and I'm sure I'm not telling this group anything new, but there is a lot of work that needs to be done to develop the social license that you need for this kind of restoration. And it really does involve a fairly exhaustive process of talking to, building relationships with, you know, hopefully including members from communities like the boating community that have legitimate concerns. I mean, these are legitimate concerns. They're not necessarily realistic concerns. I don't think, you know, a foot of elevation off the bottom is necessarily going to upset a boater, but it's a legitimate concern. And, you know, the process of physically building a reef and getting oysters onto it is generally, does depend a bit on where you are, but generally the easy part. It's the people. In fact, I have a brilliant quote from Stephanie Westby, the NOAA lead for the Chesapeake Bay oyster work. And her quote that I wrote down when I heard her say it was, "The oysters are easy. It's the people that are hard." You know, it does take a lot of effort and time to develop those relationships, educate groups on what it is you want to do, why you want to do it, why it's of benefit, and understand their concerns and work with them. So, I mean, the easy answer is there are plenty of areas that are, you know, deep enough to be out of the way of boat keels. And there are plenty of areas where if there's any boat going there, they're in trouble long before they hit an oyster. And they're the easy places to start. But, you know, that's sidestepping the work that really does, to a large extent, determine success in a lot of projects, which is how well you've socialized what you're doing. And on that score, I mean, you already have as partners the gold standard in terms of communication. The Billion Oyster Project, there isn't a project that does a better job of their public communication. I mean, the amount of oysters in the water that Billion Oyster Project have put in the water would be just a tiny dot in one of those polygons in Harris Creek, you know, where there's 350 acres just in the one creek. But nobody does a better job of that public outreach and education than the Billion Oyster Project.
Yeah, because I'm part of the community outreach team of the City Island Oyster Reef. So, you know, I come across that quite often with boaters and yachters here.
Maria, I wish I had a magic wand that had fixed that problem. It does take a lot of work, unfortunately.
Yeah, thank you.
Yes, I have a question. Where have you guys done your work, and what areas were the most? What areas were the oysters were? And I've seen them underwater when diving. So I want to know, where have you guys done work and stuff?
Okay, well, I've personally built reefs in China, Hong Kong, Australia. I've been a part of projects in Germany, Netherlands, England, and a whole stack of areas around the US. And that includes a number of different species and lots of different geographies and lots of different considerations. So to think about Eastern oysters, know your environment. Site suitability, again, is the answer to that one. A reasonable example is in Rhode Island, you get oysters from relatively high intertidal down into quite deep water in places where you have that variable salinity, where you have a freshwater input, which helps with your recruitment process. If we take that a little further south, on the Atlantic coast of Chesapeake, the oysters are exclusively intertidal. There is a predation pressure on the very small spat in the subtidal areas that don't dry at low water springs that is so high that there are no oysters surviving below low water springs. If you hop over that tiny little thin strip of land, which is the Delmarva Peninsula, and go into Chesapeake Bay itself, at that same latitude, there are no intertidal oysters. They are all subtidal. So it varies a lot depending on where you are and what the ecology looks like, what the predation pressures are, what the competition's like. And it really does, if you don't have, you know, the history of where oysters were in your area or oysters there to look at now, it does take a little bit of experimentation, just some small pilot projects to see what works and what doesn't. Sorry, that didn't answer your question, but the answer is it really depends where you are.
Thank you. I appreciate.
Dr. Hancock, I just wonder, would you mind telling us a little bit about Project SOAR? And is that a project that will be ongoing until the pandemic is finally resolved? And is that a program that we, City Island Oyster Reef, can tap into?
Okay, so I can give you a quick history actually, which does answer some of those questions. It was, Sally, I think you saw the presentation I gave to a group in Connecticut.
Mm-hmm.
It was following that presentation, a colleague from Pew got in touch and said, you know, we really should think about how we can ramp up some of this work. And we started talking fairly quickly about the problems with aquaculture and COVID. This was in April last year. Restaurants had shut down. At that point, I had no idea how long this was going to go on. I wouldn't have considered that it would be still a huge issue, you know, now. But we thought that if there was congressional relief for the shellfish growers, because the industry really was suffering, that money was likely to be handed to a federal agency, and that federal agency would need help. So we really should go about trying to figure out the information that an agency would need to be able to make a program that we envisaged work. And that program was, okay, well, these oysters, if they're breeding, they're adults, they're fecund, they're producing a huge amount of larvae, we can use them on restoration sites, particularly in the Northeast from Maryland North, where recruitment tends to be less reliable. You don't get a strong recruitment every year. So building up that breeding population. And that becomes a matter of matching up where the oysters are being grown with where the restoration sites are, because you can't just move anything around the ocean willy-nilly. That's how we wind up moving diseases and invasive species. We started putting that database together, started talking to people, and that was when one of the private philanthropy groups was actually speaking to NOAA. And NOAA said, if you have COVID relief funding, this is an interesting idea. So the program that we're running now, the SOAR program, is actually privately funded, and it's funded with $5 million. So it's a huge gift from private philanthropy because it is designed to help both the aquaculture industry and the environment, the restoration and the habitat that we're putting in. That's a finite amount of money. We have had such huge success or positive feedback, I guess, from that program, that we are looking at ways that we might be able to continue it. You know, not decided or determined yet, but certainly ways to continue it. And the other thing I should say is that there are two components of the SOAR program. One is we've got $2 million set aside to buy that 5 million oysters from the growers to go out onto restoration sites. There's additional funds to go through to cover the expense of the trucks, the boats, the barges, the pathology, the saline brine dips, you know, depending on what it is and where you are and what the invasive species concerns are. But we've also set aside $1 million for a resiliency fund. And this is looking at what can we do to make sure that the industry doesn't find itself in the same position in a few years' time when there's another, major disruption, hopefully not a global pandemic. But who knows? How do we build resiliency into the industry? And one of the ideas that is very attractive is how can we formalize this purchase of oysters for restoration, even if it's taking the seconds, the doubles, the uglies. The New Hampshire TNC program had actually in 2019 started this by buying the uglies that the industry was having trouble selling. We don't care what they look like. If they're breeding and they're going onto a restoration site, they're great. And we can get hold of those cheap. And the New Hampshire program wanted to get a mixture of age classes onto their reef to build that fertilization success. So I am hopeful, I'm not sure that I'm optimistic yet, but I'm hopeful that we will figure out how to structure a program so that this market for oysters to restoration is something that we can continue. And whether City Island can be involved in that is, I mean, there was quite a lot of activity only a few weeks ago in New York, most of it Long Island Sound. We'll have oysters going into the Billion Oyster Project. I think there's a Governor's Island site that oysters will be going into, but that's in a few months' time. Most of the New York work in the last month or 6 weeks has been Great South Bay and Long Island Sound and right out on the East End. I don't know if that answers your question, but
You did.
yep, it's a matter of the biosecurity constraints around moving oysters from one place to another and trying to match up that movement as effectively as we can.
Thank you. Any other questions out there?
I've got a question, if it's okay.
Of course.
I was wondering up here up north in the New York area, when you're trying to establish some kind of reef, would you say we have to keep the oysters in the water all the time, or how much will a freeze affect the oysters? Up here up north.
So it's an interesting question, a really good question actually. Oysters are a fantastic model for marine restoration because they're tough. I mean, when you compare them to any of those other habitats, definitely the corals, definitely the seagrass, but even salt marsh and mangroves, oysters are pretty resilient. They can handle a huge variation in conditions. I tend to think about places north of Cape Cod, even just the north side of Cape Cod, Wellfleet Bay, those kind of areas which have a lot of oysters. The aquaculture industry there, which tends to grow in the intertidal, they have quite big tides, take their oysters off that intertidal during winter. But it's not so much because the ice would kill the oysters. It's because as that ice goes up and down with the tide, it will crush a percentage of oysters, not huge, but it does damage if you're an aquaculturist and you want to sell them. The biggest damage is that oysters will stick into that ice at low tide, and as that ice lifts up at high tide again, it takes with it all of your oysters, and who knows where they go. So it's not necessarily killing the oysters, but it's not doing you any favors. So in that sense, sea ice, pack ice is certainly something that is best avoided. I don't picture that you have that kind of issue around City Island. I mean, a bit of ice, they can certainly handle. You would also be thinking about, poaching. Are you making these more attractive by putting them above low water springs? Those kind of issues. But yeah, they're definitely sensible considerations.
Would a week of 20-degree weather hurt the oysters?
They survive in the intertidal quite happily north of Cape Cod. So on a species scale, no. I would be careful with that answer though, because local adaptation actually is real. It is measurable. There are differences in genetics of your local stock between areas, and I think it would be well worth avoiding. My advice usually is when you're first starting to restore any of these habitats, particularly oysters, do it in the place that's most likely to give you the best result. Go to the best spot first, and that's inevitably where the oysters disappeared last. Where they hung on longest is a very good indication that conditions there are the best. But yeah, if there is a percentage mortality from a long cold snap, try and avoid it. Even if the oysters can survive in those conditions, it's not where you want to put your first project.
I think Rob Craffa had a question. Rob was put on the chat that he was wondering, oyster drillers are a real problem in this area. Rob runs the maritime waterfront at SUNY Maritime and has a lot of oyster cages and very involved in it there and out in Oyster Bay out in Long Island. But in this area, they're particularly problematic. And is there anything that can be done about oyster drillers?
Yeah, that is a difficult one. It's like some of the diseases. Once these invasives are a part of the ecosystem, they are endemic. There's really no getting rid of them. Unfortunately, things like derma we've had to figure out how to live with rather than how to avoid. And that's certainly the case for the oyster populations. And it really does make the inertia of getting started heavier or harder. It does. One of the most difficult things, I think, is scale. And that's not in the sense of how much is enough, but how big do you need to go to start to get a result? Because, that kind of predation, pressure, and competition does play into the scale argument. Do you need an acre of high density oysters so that there are enough oysters to feed the drills and have a reef persist? Is a 5 square meter patch just going to be swallowed up in a week? They're questions that I'd need a lot more very local knowledge from your area to be able to answer. But they're the kinds of considerations that are very important.
How about the size of the oysters? Do they resist the oyster drills at a certain size, or they're susceptible at any size?
Your mortality generally goes down quite fast. So for the Rhode Island study that Sally mentioned at the beginning, spat on shell that I was putting out, I was getting a 75% mortality. For the next year, so for the second year that they've gone out, it's only 25. So it goes down very fast. The bigger they are, there is a size refuge, they call it. The bigger they are, the harder they are to break into. And that's for drills, crabs, turtles, anything. They do become much more resilient the bigger they get. Their highest mortalities are definitely when they're small.
Interesting. Then if we held, we're raising the oysters from they started about 12 millimeters. And in the season, they're growing to about 50 millimeters in one season. Would it be better if we didn't put them out after the first season, or tried to overwinter them and keep them off the bottom or keep them away from the predators, grow them one more season on top of the water in cages, and give them 2 years before we put them out?
So in the long run, to get to any kind of ecologically meaningful scale, you're going to have to be moving away from that intensive husbandry, because it just takes too much time and is too much work and equipment and all of the expenses that go into that. But to get reefs started, that is certainly an option. And lots of projects have done it, and I've done it myself, and it can be quite successful to look after those oysters, to get them to a size where the mortalities are low enough that you start to see a reef persisting and building rather than dying slowly over time. Unfortunately, I can't give you a general kind of modeled answer to that. My best answer would be that's a really constructive experiment, and that's a really well structured experiment that I suspect you'd be able to get a university interested in. Now, that's a great student project.
Yeah,
You might-
if I could have a question along those same lines, that's quite beautiful. We have a 5-mile opportunity from David's Island down to Throg's Neck. And it seems like, just as per what you just said, we have basically, and we have a kind of model for this, how we could have essentially lines of hard substrate structure going down a meter wide, doesn't matter what the width is. But do you see an opportunity to basically, who would we draw into that on the university scale that you know about? Because it's a kind of a regional scale opportunity to look at all the questions you bring up, and I'm just wondering who would partner with us on that front.
Wow. First name that comes to mind, I mean, there are certainly options for universities in your area. One of the advantages is you have to have universities in your area. Stony Brook is, Stony Brook's
Well, I
very
know
involved in
Jeff Lovington very well. It's just a matter of, I'm looking for, is there anyone who, if we said this to them, they would call us back? Who's on a structure, because we would love to incorporate graduate students, postdocs under project opportunity is enormous, but you need people who love it as you obviously do, but somebody who wants to get in the water basically and get their hands in with us to see what can happen.
The advantage of a student is they've got a degree riding on them doing a decent job of this, so there's focus.
Yes, indeed.
My first response actually would be Stony Brook, but
I literally just sent Jeff Lovington an email yesterday about the scale that we would need for what you described, dropping down anoxia with filter feeders. So I'll see. I just if keep it in mind, we would love to actually partner, and the scale, as you say, it's critical because a patchwork is always going to be in trouble or potentially, but getting this kind of reach, because that's what could make a difference. Just I loved your beautiful line crossing with the population and the water clarity, and you didn't correlate that directly with fishery production, but there it is. And we want to show some of that, and I'd like to measure the pieces going forward. So we can certainly go with Stony Brook, but any collaborator that comes would be good for us.
Yep, well, certainly you've got the right ideas there, and you've already written to my first suggestion. So I wish you luck. Look, if you don't have any success there, I'd be happy to think through options and ask some of the network. We have a TNC Long Island chapter that's quite involved in some of this work. They've probably got ideas as well.
Yeah, thank you.
Well,
Boze, if you got a second there, so a lot of times when I'm diving, I'll come across like 7-inch oysters, 6-inch oysters. They're not really clumped together. They're kind of scattered apart. And as much as exciting that looks and feels, I've been told by other divers, other guys that have seen it, that it's not an area of concentration that we really want to look at. We want to find some place with a more populated density. What's your thought on that?
I mean, it's not uncommon that you get better recruitment than you're expecting because you don't see many oysters around. Those odd individual oysters don't make up a habitat. They're not influencing, you know, the ecosystem services on the scale that have an impact. But those big old oysters tend to be female and they produce a lot of eggs. So as long as there are enough to have effective fertilization, they can be really critical in providing recruitment for substrate that goes out. If you're building a reef, even if you're putting spat on shell on it, it's not uncommon to come back, you know, in your first year and find babies settled onto your spat on shell, and they have to have come from somewhere.
Right.
It's those isolated oysters that are doing that. At the same time, you know, one of the common targets or success metrics for reef restoration is a variety of age classes. These, they call them a protandrous hermaphrodite. Mollusks are amazing. You know, they do whatever they feel like at the time with sex. It's you know, they can be male, they can be female, they can swap fairly regularly. The oysters are certainly capable of going both ways. They tend to generally mature as male, so the smaller ones tend to be more male, and those large ones tend to be more female because they've changed over time. So just having a mixture of the large and the small, a mixture of age classes, is one way of guaranteeing good fertilization success during spawning. That's one of my concerns when you start finding the odd big oyster scattered around, that there aren't enough of them close enough together to really create a successful spawning. But at the same time, we do fairly commonly see better recruitment than expected. So, you know, it's working. You know, I'm not sure quite how to answer your question, except to say that, you know, I can't help myself when I'm diving. I tend to grab those big old ones and put them in a bunch of a dozen together.
Yeah.
That's getting a more guaranteed fertilization result.
Right.
It's not building habitat in the sense of an oyster reef. That takes, you know, the process that we've described, the substrate
Right.
and some structure,
Yeah.
etc.
Yeah,
But
that
it
was kind of my thought was, you know, the closer they are together, the better they were going to survive.
Yep, absolutely. Well, it also speaks to that question about survival too. It's those big old singles that, you know, are living despite everything else because they're big enough and tough enough to do it.
Yeah.
And there's only a few of those baby spat that have made it to that size.
Yeah. Yeah, that was kind of like my thing, was feeling like maybe fill in substrate in between them, you know.
Yeah, I tend to pick those guys up, and when I find something hard enough that they're not going to sink into the mud, I'll put them together on that.
Okay.
It's kind of probably more of a feel good exercise than
Yeah.
a functional exercise, but I can't help myself.
Yeah.
If they're in the floating cages, will they lay their eggs or do their spat, and will they fertilize amongst that floating cage?
Absolutely, yes. So, you know, the ecosystem service, it's an interesting field that TNC is getting quite active in, actually. We call it restorative aquaculture. So it's the ecosystem services that make these reefs hugely beneficial. Oysters in an aquaculture setting do many of the same things. The services they provide are going to be in different amounts. So you may not get nearly as much fish production from oysters in aquaculture gear as you would from an oyster reef. Maybe you will, and it's certainly structured habitat. That's something that we're trying to measure. But all of those oysters are breeding. You know, by their second year, oysters have matured. So even a 2-year-old oyster, not producing nearly as many as eggs as some of those big guys that you're finding as singles on the bottom. But in aquaculture settings, there are a lot of them. So yes, they are certainly spawning and producing larvae.
Almost like breeding bags in a sense.
Yeah. Yeah, there's actually been a study here in Rhode Island that's correlated quite neatly that aquaculture broodstock with increased recruitment on the bottom.
I have another question. So when you were showing the ecoregion map in the beginning,
Mhm.
I noticed that one of my favorite places to visit in the entire world was Africa, and there was absolutely no shellfish restoration projects going on there in the entire continent. And I was wondering why that is. And then the other place I noticed that there were some very, very successful projects going on was in South America. And I was just wondering which regions in South America were the really successful ones.
Okay, so that was not actually mapping out restoration as much as mapping out the habitat that exists or remains globally. And it was constrained by A, where we could find information, and B, where we could find somebody that had access to that information. And I actually spent a lot of time during that study. I thought there must be people in South Africa particularly that had that kind of information, and I didn't uncover it. Now, I was in South Africa in late 2019 and was talking to the kinds of people that I wish I had found in 2008 when I was starting to look for them in South Africa. That information does exist. It's harder to find. There are oysters around the African continent. We just in that study didn't find the people that had that information in the structure that we needed to be able to publish it. So there are lots of gaps on that map which could be filled in. And actually one of the nice things about that historical ecology is that when you start, you have no idea where this information is going to come from. And the more people you talk to and the longer a project goes and the more interest there is, the more information comes to the surface. So, you know, in the 10 years since that study was published, there has been a lot more information that's been brought to the surface.
And where are the places in South America where they were very, there was a lot of.
Okay, so there were two patches on that map that were above 25% remaining. One of them is actually a fished area south of the Amazon mouth in Brazil, where you've got a mangrove oyster. And it's a traditionally fished resource. It's quite an important cultural fishery, and on a family or village basis, it's quite strictly managed. They have figured out over the hundreds, if not thousands of years, how to fish those oysters without killing them. So it's actually a really interesting example of some of the local management and the potential for that local management. So that's a fairly discrete area of Brazil where there's a mangrove oyster. There is some restoration being done in South America. We've been involved in some in Mexico, but it's fairly patchy down as far south as Uruguay. Actually, the other green area on that map was a patch of the Gulf of Mexico, which unfortunately has also changed quite a bit. You've probably heard of Apalachicola was in that area, and that was a big fishery in 2010. That is no longer. It's been shut down. They've killed it off nearly completely. It's another restoration prospect.
Thank you.
Dr. Hancock, I want to thank you. I know it's been a lengthy evening. It's been a fascinating overview of what's happening in the world with oyster reef restoration. Really, on behalf of all of us from the City Island Oyster Reef, and for all of our advisors who are also here representing SUNY Maritime, Billion Oyster Project, Leah Hill is here from the Nantucket Shima Creek. We all so wholeheartedly thank you for, sharing your time and your knowledge about really what's happening in the world and throughout the United States with oyster reef restoration, and all the photographs give such a concrete visual. of all the different types of construction that's happening. And thank you from the bottom of our hearts. You took
Thank
my
you very
call
much.
and you came here, and we all really, really appreciate that.
Well, no, thank you very much. Keep up the good work.
I think when the pandemic's over, I think we got to take them out to eat for a good seafood dinner. at City Island.
Definitely.
Yay.
Absolutely. We'd love that.
Yeah.
Bring on the end of the pandemic. Absolutely.
Exactly. We
Thank
would
you
love
very
that.
much.
Okay. Well, thank you.
Thank you.
Good night.
Thank
Good
you.
night.
Bye. Thanks so much.